Hello, everyone. I'm Tiana Ellington. I'm the Assistant Director of Chapter and Regional Programming with the Alumni Association of Case Western Reserve University, and it's my pleasure to welcome you. Before we begin tonight's program, in conjunction with the School of Dental Medicine, I have one housekeeping note. Everyone is muted by default in an effort to limit background noise during the your loved ones are healthy and safe, and I have a few campus updates. In response to the COVID-19 pandemic, Case Western Reserve's research has become vital to the fight, and we invite you to follow us on Facebook and the university's website, case.edu together, for regular updates such as this event we will be starting momentarily. In support of the ongoing conversation about race, equality, and social justice in our country, Case Western Reserve has launched the website case.edu/raceandjustice, providing statements from university leadership and university resources, as well as the opportunity for members of the community to share their thoughts. You can also find more information on the site about CWRU's second day of dialogue hosted on September 17th, where the community worked to continue conversations related to these critical topics and drive action. At the Alumni Association, we continue to use our platforms to promote connections within the CWRU community and present information and opportunities that are available to all. For regular updates and to connect with the community, follow us on Facebook, Twitter, Instagram, and LinkedIn by searching at CWRU Alumni, and visit our remote offerings page at case.edu/alumni for a collection of these opportunities. Now more than ever, we hope these virtual events and social media platforms will help us continue to stay connected. Again, thank you for being a part of this evening's event. We appreciate you. And with that, please allow me to welcome our facilitator for this evening, Dr. Fadi Fadul.
Thank you, Tiana. Good evening, everyone, and welcome to this evening's event. My name is Dr. Fadi Fadul. I am a professor and chair of the Department of Comprehensive Care at Case Western Reserve University School of Dental Medicine. It is my distinct pleasure and privilege to introduce our renowned speakers this evening on a newsworthy and timely topic of the SARS-CoV-2 virus and its impact on animals. The COVID-19 pandemic and human-to-human transmission of the SARS-CoV-2 virus has been the focus of worldwide attention and ongoing headlines in global news. We are beginning to have a better understanding of this complex virus and modes of transmission. However, a new list of potentially infected mammals is only beginning to receive its deserved attention. That list includes the 240 mammals described in a new paper published in the Proceedings of the National Academy of Science, spearheaded by Dr. Harris Lewin and Dr. Joanna Damas at the University of California Genome Center, and joined by two co-authors this evening who will share this work, as well as two additional speakers who can give us a larger frame of understanding on the subject. Before I introduce our first speaker, I want to encourage everyone to put their questions in the Q&A box that is at the bottom of the screen and throughout the presentation. We will be answering the questions at the end of the presentation when all speakers have completed their presentation. Thank you again. And now please allow me to introduce our first speaker, Dr. Martin Nguyen, who is an assistant clinical professor at CWRU School of Dental Medicine in the Department of Comprehensive Care. He is a lecturer at the Harvard School of Dental Medicine and research scientist at the Smithsonian Institution and Canadian Museum of Nature. Dr. Nguyen.
Greetings, and thank you, Dr. Fadul, and the organizers of Case Western Reserve, and to you, good evening, good afternoon, or good morning, whichever your time zone might be. And welcome to this seminar based on the COVID potential transmission to wildlife. The focus of today's presentation begins with a recent publication, as Dr. Fadul mentioned, in the proceedings of the National Academy of Science. And this was created and started by biologist, immunogeneticist, and one of today's most innovative scientists, Dr. Harris Lewin, powered by Joanna Damas, the engine driving the data. Dr. Oliver Ryder and myself, two co-authors from that publication, are here to present aspects of that study which have importance in the natural world and to our research interests. Now, many insights have arisen during the pandemic, and some of us see it as an opportunity to care, listen, and remain connected to our planet and the 4.5 billion years of knowledge that she holds. Yes, we have 1.1 million deaths and 41 million cases of COVID reported worldwide. But we also have 2.2 million deaths each year just from drinking contaminated water, according to the World Health Organization. The 2019 World Drug Report from the United Nations Office on Drugs and Crime states that nearly 750,000 people die each year from drug-related overdoses. The SARS-CoV-2 virus will be our main focus, but it represents a broader vision about how we live in and connect to our planet. So we've read and seen on the news reports of human-to-human transmission of COVID, and now our animal populations are catching up, with the culling of 1.2 million infected mink in the Netherlands recently, and more than 750,000 in Spain. Other species already infected include several lions and tigers at the New York Zoo, and small numbers of dogs and cats. Research on small groups of non-human primates and other animals shows viral infections of those animals and transmission of infection to other animals of the same species. Certainly, animal-to-human transmission was the initial path for the spread of this virus, but our work here involves human-to-animal and animal-to-animal pathways. So to be clear to indigenous audiences, there is little to no risk in consuming country foods. This investigation is about how we as humans may impact animal populations and how animals can then spread the virus potentially to other animals. So how does this virus infect humans? Well, science has shown that the COVID-2 virus binds to the ACE2 cellular receptor, which is commonly referred to as angiotensin converting enzyme 2. Now, this is a protein that functions mainly in the cardiovascular system and is important in regulating blood pressure. Because the ACE2 receptor is found in all vertebrates, we examined 410 species to see if there were similarities in binding sites that would serve as a host site for the COVID-2 virus. So, among those groups that shared all 25 of the ACE2 binding sites with humans were old world primates like chimpanzees, orangutans, and gorillas, labeled in this case as very high risk. The second group of high-risk animals that shared 22 of the binding sites on the ACE2 receptor were narwhals, which I have studied for many years, and other whales. So, you're probably wondering at this point, how did a dentist end up studying narwhal in the high Arctic? The narwhal tusk is the most extraordinary tooth on the planet and defies almost every principle and property of tooth expression I ever learned when I was a student at Case Western Reserve. So I decided to venture nearly 2,000 miles due north almost every single summer for the last 20 years, donning a dry suit in 36-degree water to do physiologic research and assist on tagging research teams with Fisheries and Oceans Canada, and also collecting traditional knowledge along with anatomic specimens. So we know that the Arctic environment is changing in multiple aspects at a rate of 2 to 3 times faster than the rest of the planet. Changes to the sea, including loss of polar ice sheet cover, CO2 absorption, ocean acidification, and alterations in the chemistry of calcium. Changes on land also are dramatic, including animal crashes, like the caribou population dropping from 5 million to 2 million in just 2 decades. Studies on animal populations are continuing under the Smithsonian Arctic Crashes Project. Even the atmosphere is changing. Magnetic north, you might think, is a still point, but it's actually moving at the rate of 34 miles per hour toward Siberia. So fast that the world magnetic model, which guides everything from navigation systems on ships to smartphone applications using the Google Maps, needed to be updated last year. So variables like animal migration, seasonal change, and ice and weather that give meaning to the 6-season Inuit calendar are all in flux, making an already challenging lifestyle more unpredictable. Now, of the 252 mammals examined in the paper, toothed whales are in the high-risk category for binding the SARS-CoV-2 virus and contracting COVID symptoms. So what does that study actually mean for Arctic species? Well, a Stanford study looking at toothed whales like narwhal and beluga found that they didn't have a necessary gene to adequately establish viral immunity or mount a defense against many viruses. Now, certainly there must be some advantage for these whales to have ACE2 receptors, since they are positively selected for some reason. One important piece involves obviously blood regulation for these deep diving whales. But based on high risk for binding the virus and the lack of necessary immunity for toothed whales, at least one we understand, more study is needed to establish whether this risk can involve infection in the wild and examine the possible modes of transmission that might cause infection. Don't mean to keep a waste treatment plant on your screens for too long, but if you follow through this next paragraph, research up to this point has focused on respiratory transmission. But we also need to consider wastewater and fecal contamination. The SARS-CoV-2 virus can survive up to 4 days in fecal material and over a week in wastewater. Two recent MedRx papers cited detectable fragments of the SARS-CoV-2 virus in the wastewater systems of 7 European cities weeks before the COVID outbreak. And likewise, Monitor of Boston wastewater titers found the SARS-CoV-2 virus as well. So even if there is not a direct link to active infection, sewage detection may be a good indicator of what viral infections might be coming. Contamination can occur in multiple ways, giving increasing boat traffic, wastewater runoff, and substandard water treatment facilities. Though speculation might lead to the assumption of immediate destruction of such viruses in the Arctic Ocean, we need to understand a little bit about the Arctic Ocean. First, it's the world's least salient ocean, with multiple freshwater rivers flowing into it and the contribution of rainwater via ice melt. In addition, there are multiple brackenwater inlets used by migratory marine mammals, as well as glacial runoff and summer ice melt, which add to the freshwater system that can affect this marine life as well as the virus survival. A recent study from the University of Colorado at Boulder indicates that since the 1990s, there has been a 20% increase in freshwater areas in the Arctic Ocean, totaling 10,000 cubic kilometers. That would roughly equal a depth of almost 1 meter if spread out evenly across the entire Arctic Ocean. Our freshwater systems are in peril, and much of the world does not have access to safe drinking water. We mentioned earlier the 2.2 million people dying each year of infectious diseases spread by contaminated water. Our oceans are also suffering. A recent Nature article states that viruses in the sea represent a reservoir of the greatest genetic diversity on Earth, with a population of nearly 200,000 different viruses in the world's oceans environments. Most of these have restricted hosts and pathways, but the important message is that there is a vast viral ecology that we are mixing into an ocean soup with waste products and a planet that now has 7.8 billion people. Now, to date, coronaviruses have been found in beluga, bottlenose dolphins, and harbor seals. And viral pandemics are real for marine mammal species. In 1988 and 2002, a breakout of the distemper virus in harbor seals killed over 50,000 animals. So the potential risks are real as we continue to batter the environment with collected waste and fecal material at one end and increase our consumption of everything from oil, natural gas, minerals to food and building materials. Our studies this year will continue to investigate live narwhal cells and their capacity to bind the SARS-CoV-2 virus. We are working very closely with our next speaker, Dr. Oliver Ryder of the Frozen Zoo in San Diego, to better understand the predictive table risk for this animal susceptibility to this virus. Keep in mind, the table that was established in this paper for the National Academies of Science is a predictive table. To make the reality of these predictions, we actually need to get live cells from some of these animals and discover if indeed the SARS-CoV-2 virus has the capacity to bind to them. As with all good, provocative, and thoughtful research, insights from the pioneering work of Dr. Lewin, Dr. Damas, and other collaborators give us answers that can also lead to better questions. Inuit subsistence hunters are safely consuming their hunt, but will we need to expand the monitoring of potential animal contamination? And what about waste and fecal contamination? How can we monitor new types and increasing amounts of human waste associated viruses in the marine environment? Now, I don't know where you folks live, but I'm from Connecticut originally, and we had a power outage 3 weeks ago. During that power outage, one of the most progressive states, Connecticut and Massachusetts, had power outages that caused backup generators at their waste treatment plants to fail. That dislodged a little over 1 million gallons of untreated waste into 2 main river sources in our states. So you can imagine what happens in progressive states. Now think about what happens in unmonitored areas. This is the kind of work I'm doing in the field. I just wanted to give you an illustration that when I said I was donning a dry suit in 36-degree water, that it's actually real. And this is the location of the studies we do. We're in the high Arctic. The samples that we collected for Dr. Oliver Ryder this summer were in Pond Inlet in the map to the right. We all know that plastics have already made it into the Arctic food web. So should we not be looking at viral contamination sources? This viral pandemic should be a wake-up to all of us to let the science inform us about how we live, care for, and respect our planet and our shared home. Thank you very much.
Hi, I'm Dr. Oliver Ryder. I work at the San Diego Zoo Institute for Conservation Research. This is the Beckman Center for Conservation Research, where our laboratories are and are home of the Frozen Zoo. San Diego Zoo operates the Institute for Conservation Research. It's the largest zoo-based multidisciplinary conservation science effort in the world, as far as we know. A prime reason for me being here to talk to you is because, as Dr. Nguyen said, we have what we call the Frozen Zoo. This is a collection of living cells that are propagated from small biopsies that can be obtained from living animals or animals shortly after they're deceased. The cells can be grown in the laboratory and then frozen in liquid nitrogen and kept for a very long time. We've been thawing cells out recently that have been frozen for more than 30 years. The Frozen Zoo has cells from over 10,000 individual vertebrate animals, terrestrial vertebrates, mammals with backbones that live on the earth or in the air above it. We have, that's over 1,000 species, and the image shows a breakout of the number of individuals. Most of them are mammals, but the most rapidly growing portion of this is now bird species. One of the uses of these frozen cells that contributes to conservation is to sequence their genomes, to use high quality samples as that can be obtained from cells to produce the highest quality genome assemblies. The Frozen Zoo has contributed very substantially to the development of the field of comparative and conservation genomics through projects such as Genome 10K, the Vertebrate Genome Project, and now the Earth Biogenome Project. So the rate of sequencing genomes is expanding very dramatically, especially including genomes of new species, but it requires having a suitable sample. And that's where we are able to make contributions, because we've been saving cells in the Frozen Zoo since 1975. Because of the number of genomes that have been sequenced, that allowed the paper that Dr. Nuia has mentioned to be developed, because the data were already available from hundreds of mammalian species to do the estimation of the risk of binding of SARS-CoV-2 virus with cellular receptors, ACE2, across the wide variety of animals. And as Dr. Nuia showed a picture of a gorilla, they're estimated to be as prone to risk of infection through this ACE2 receptor by the SARS-CoV-2 virus as are humans. One of the surprises, though, as he mentioned, was that this study led by Joanna Damas and in Harris Lewin's group identified other species that were at high risk of infection using the methods that were developed for this study. And it included cetaceans, whales, but we had very few whale genomes to analyze. So that's one of the reasons it's very important to actually get samples from narwhal, grow the cells, and allow the further identification of risk across a larger number of species. And this effort is in fact ongoing. There's over 15 species of cetaceans, whales, and dolphins in the frozen zoo. And this is probably the largest collection of cells from this group of mammals of its kind. And as I mentioned, it's really important that the narwhal be included. But we need to find out about other species of whales. The largest animal on Earth is the blue whale, and it's a recovering endangered species. And the marine environment is hospitable to the transmission of these viruses, it seems likely. So this is important to address. We were able to establish cells from the most endangered cetacean, probably one of the most endangered mammals, the vaquita, which is a small dolphin porpoise, and establish cell cultures from that, which has allowed a genome assembly that can be used for predicting its extinction risk as a factor of identifying the level of inbreeding or deleterious mutations that are in small populations. This is part of the reason that we use the frozen zoo. But we've been able to also use advanced reproductive technologies in the frozen zoo to produce clones, to produce copies of animals that died and their cells were in the frozen zoo, and then they had a chance to live again. These are two relatives of cattle that were among the first endangered species to be using cloning technology. And just in this last 5 weeks, we reported in a collaboration with a nonprofit, Revive and Restore, and with Viagen Equine, the cloning of a Przewalski's horse that's living in Texas right now with its surrogate mother. So the uses of the frozen zoo are enormous and expanding, and it really was a well-placed decision in 1975 for San Diego Zoo to start this effort. And one of the things we're doing right now is trying to save one of the most endangered mammals, the northern white rhinoceros. There's only 2 females that survive, but we have cell cultures from 12 northern white rhinos in the frozen zoo. And we've been able to make pluripotent stem cells from these with the methods that that preserve the integrity of the genome, so that we could actually we would like to direct these stem cells to produce gametes, to produce eggs and sperm, so that we could actually have the cells have offspring. And as an example of the potential of these cells to undertake all of the functions that take place in a developing embryo, here in the video is a slide of beating cardiac cells that are from a northern white rhinoceros. that died some years ago and whose cells have been in the frozen zoo. So we regard this as a hopeful enterprise to make use of advanced cellular and genetic technologies. We didn't anticipate all of these applications in the 1970s when when the frozen zoo was being established, and there was 10, 20, 30, 40 individuals banked, and the number of species was accumulating up to the more than 1,200 that we have now. But it stands as a very valuable contribution for the future, and we're delighted that it's been able to contribute to a fuller understanding and prediction of SARS-CoV-2 risk infection across from humans to other animals and between animal species that are especially, that could have their populations reduced or potentially suffer severe consequences because of this pandemic infection. So thank you for your attention, and I'll be happy to answer questions at the end of the time.
Thank you, Dr. Ryder. I apologize. I couldn't unmute myself to introduce you, but I'm going to do it now, if that's okay. Dr. Oliver Ryder is the Kleberg Endowed Director of Conservation Genetics for San Diego Zoo Global, and adjunct professor in the Department of Evolution, Behavior, and Ecology at the University of California, San Diego. Thank you, Dr. Ryder. It is my pleasure now to introduce our next speaker, Dr. Judy St. Ledger, a veterinary pathologist, former Vice President of Research and Science for SeaWorld, and current adjunct professor at Cornell University. Dr. St. Ledger.
All right, now this is Dr. St. Ledger. And we're going to wrap up with Dr. Bird, I think. And I am a little bit different from this team in that I'm not on the paper that we're talking about this evening, but I am a veterinary pathologist. And I'm a veterinary pathologist who focuses specifically on diseases and concerns of wildlife. And if you know me, you know that I focus amazingly on diseases and concerns of wildlife, specifically marine wildlife. So what I'd like to do is just spend a few minutes and talk a little bit about the role of wildlife in the SARS-CoV-2 disease. And I will tell you, I started this presentation by talking about zoonotic disease associated with SARS-CoV-2. Then I talked about what we call anthropogenic disease or reverse zoonoses, which is diseases animals get from people. And then I sort of became enlightened, and I decided we'd talk about how diseases are shared between people and animals. So the first picture here on the left is a picture of the Wuhan wet markets, where you can see lots of wildlife, both intact and as meat, that are available for exposure as well as purchase to people. But here on the left, we see a picture of a man kissing his kitten. And you may say, my gosh, what the heck does kissing a kitten have to do with lizards that are available in the Wuhan wet markets? And I'll tell you, it has everything to do with them. Because what it has to do with them is the broad host range of this coronavirus. So is everybody on board? Oh my gosh, it's not just lizards, it's not just bats, it's everything including kittens that we have to consider when we think about coronavirus. Now, I'd like to take a few steps back and talk to you about how people interact with wildlife. And certainly, I think everyone on this Zoom can agree that we have increased interaction. So one of the ways we have is increasing human habitat or encroachment. And for a long time, I lived in Southern California, the world where a great house is one where you could have deer in your backyard. And now I live in upstate New York, where having raccoons in your backyard is seen as attractive, unless you're a veterinary pathologist, and then it's seen as a huge risk for things like rabies and potential of other viral infections like paramyxoviruses or coronaviruses. So backyard wildlife is important, not only for exposure to people, But let's look at this picture here on the right, exposure to animals. So think about pets you have in your home and the fact that they could interact with wildlife and then they could interact with you. Talk about working as a scary intermediate. I don't know about you, but I don't think Tigger needs to be an intermediate for me to move COVID-19. Now, oh, that it were only human encroachment that was the problem. These are pictures from Indonesia and slash and burn activities to expand agricultural space, reduce wildlife habitat. So not only are humans moving into the wildlife habitat, but we're making the wildlife habitat smaller. And here on the right, we see pictures of orangutans who have lost part of their habitat because of slash and burn agricultural activities. And all of this goes to putting people in direct contact with wildlife. Now, remember, when we talk about emerging infectious diseases, we're concerned about the fact that as we increase our exposure to wildlife, the transmission of these viruses can become more significant. Well, we've got them in our backyard. We're moving into their backyard. My God, what else can we do? Well, I'll tell you. The other thing we can do, and this speaks to the sort of things that Dr. Nguyen talked about, is we can have global climate change or ocean temperature changes really changing the way that animals live and where they live. So in June 2020, I want to bring this back home to things we can relate to today. In June 2020, off the coast of San Diego, we identified a beluga whale. And I kid you not, I probably had 20 people that called me and said, hey, do you think SeaWorld lost a beluga whale? And the answer was no, SeaWorld did not lose a beluga whale. But a beluga whale, which should have lived in the Arctic zone of the globe, migrated. And it migrated far, down to the area of San Diego, California. Well, unfortunately, not only did a beluga migrate to San Diego, California in June 2020, but in this month, in October of 2020, the animal unfortunately was identified dead on the shores in Baja California, Mexico. And the beluga whale that had been seen in what we call an extraliminal habitat had died, unfortunately, because beluga whales don't belong off of Southern California or off of Mexico. The point of this is that climate change and changes in the ocean condition are dramatically impacting our relationship to both wildlife and to the wildlife relationship to the environment. And when we look at the interaction of wildlife, environment, and our impact in all of this equation, we find that we are setting ourselves up for increasing exposure to infectious disease. Let's talk about this ease of exposure. Well, is it all just crazy wild animals going nuts? No. What I've done here is chosen a number of pictures that demonstrate the sort of things people do to really increase their exposure to wildlife. The first is a picture from Georgia Aquarium, but it could have been SeaWorld. It could have been a number of facilities that have beluga whales. And imagine if this woman in the middle of the picture has COVID-19. We've already heard from Dr. Nguyen that what we see, and Dr. Ryder, that what we see is a highly susceptible species that is being exposed to another highly susceptible species. Dear God, could we set up an easier transmission? I don't think so. So these interactive animal events certainly are an easy way to transmit virus from people to animals. Isn't it crazy? We also could transmit it from animals to people. Keep in mind that this study that was published looked at the ease of infection in many species, and we are likely to have infected marine mammals. Now, it's not just marine mammals. Here we have a porcupine species, and these folks are directly in the airspace and interacting with the porcupine. And on the lower right, we have folks that are happily feeding giraffes in a facility in Great Britain. The reason I put this in is humans like interaction animals, interactive programs, and they really like the thought of wildlife rehabilitation. So what the heck are we talking about when we're talking about wildlife rehabilitation? We're talking about babies that are either abandoned from their moms or separated from their moms and need care. We're talking about animals that are unable to feed themselves for one reason or another, either through infection and disease or because of decreased ability to sort of act appropriately to feed themselves. But what I've done is I've chosen a series of pictures that demonstrates a seal, a raccoon, a squirrel, and unfortunately a white tiger that are available for care and need care. Is there anyone on this call that thinks that any of these baby animals don't need care and shouldn't be cared for so they can live a happy and healthy life? I hope not. But unfortunately, I hope everyone can see the direct relationships between humans and wildlife in these images. So while some of these humans have on gloves and they may have on masks, what I want you to appreciate is that as humans, we actually strive to live with wildlife in our world. And when that's the case, we need to accept the fact that infectious diseases are also part of what we can expect to experience in this relationship. It's a little scary, isn't it? All right, let's go to the next slide. All right, let's talk a little bit about the conservation of these ACE2 and these ACE2 receptors. So what species might serve as intermediate hosts? And what I'm doing now is referring back to the publication that we all are sort of thinking about, which is the host strains of SARS-CoV-2. So we have non-human primates, NHPs, that have a very high conservation. In other words, humans probably should not, have dinner with gorillas or orangutans. But what about having dinner with the cetaceans, bottlenose dolphins, or gray whales? What about having dinner with cervid deer? Should you be feeding Pierre David's deers? And what about hamsters? And we'll talk about hamsters in a minute. Now, we know that we have different rates of conservation of the ACE2 enzymes in different species. But we also know that we have very low conservation in species like the pangolin and bat species. Now, hold on. I'm guessing that folks know that bat species are a concern as an intermediate host for viruses like COVID-19. How does that work? Well, remember, if you look at this manuscript, we have some lineage-specific pressures for bats to a really specific part of the genome called Q728. And that being the case, perhaps bats really are good at amplifying a number of viruses. So you and I probably don't have direct connection with bats, but those bats may be good at sort of amplifying viruses and then helping to spread it. Where do bats spread in the night? I have no idea. I'm sleeping. And the fact is, most of us are sleeping, but the bats are out working and moving about. And it's that moving about that may be of concern and importance. Now, so which animals have had proven natural infection with this virus? And Dr. Nguyen referred to them earlier. We've had these lions and tigers in a zoo facility in New York City. We've also had identified natural infections in both cats and dogs that were identified by a number of commercial laboratories in the United States. And of particular interest are these farmed mink. And the reason they're important is when we look at the conservation of their ACE2 enzymes, it's not nearly as similar as we would think to human enzymes. So the question is, why do these mink get sick? Well, there's a few things to think about. One, the mink may become infected and not necessarily become sick. But two, we've also identified specifically in the Netherlands, I believe, may have been Denmark, not sure, that the mink were able to transmit the virus to both cats and dogs. In other words, we didn't have animal to human infection. We probably had human to animal infection. That's how the mink got sick. But what we may have had is animal, mink, to animal infection to cats and dogs. This is important because what this entire investigation is about is what's happening with the virus and who can get it and who can share it. So let's take a look at some of these experimental infections. And what we're looking at here is an image of an experimental infection in a rhesus macaque, where they're using a vaccine, obviously, to prevent transmission of the virus. So we know in a number of non-human primates, rhesus macaques, cynomolgus macaques, grivets, which is a kind of guenon, common marmosets, these species get infected and those poor animals get sick. They get sick from SARS-CoV-2. They have pulmonary infections. They can have a variety of systemic infections associated with the viral infection. Now, there are a number of species, ferrets, cats, Syrian hamsters. These species can become infected and they can spread infection, but they don't necessarily get sick. And when a number of species like mice, pigs, chickens, and ducks were infected, well, you can give them the virus, they don't really seem to get infected. So the fact is, the ability to infect different species of animals differs by animal species. And that relates directly back to this publication, which said, hey, maybe it's associated with this binding enzyme. And if that's the case, how does it all relate to the world? Well, let's talk a little bit about how we deal with these infections. And the reason I want to talk about this is Dr. Nguyen talked about those darn narwhals. He loves narwhals. Well, I'm kind of crazy about belugas. So it's similar, but a little different. My animals don't have a big tooth. But what I'm interested in is the fact that there is a gamma coronavirus that has been reported in both beluga whales and in bottlenose dolphins. And while we may know some of the binding protein concerns with COVID-19, we don't know the binding protein concerns for these other coronaviruses. And in the world of dolphins and whales, I think that information may help us to understand better what the heck is going on with COVID-19. So when you say, how do we deal with these infections in both animals and man, it's all the things we know about. It's basic infectious disease control. It's routine surveillance. It's making sure laboratories are capable of dealing with things. It's promoting and supporting research. It's making sure we have training and education. And super important, it's communication. We need to make sure that globally we have established cross-sectional networks so that when someone finds something unusual or something that seems inappropriate, they can communicate that so we're ahead of infectious diseases, not behind them. And I think that's sort of my take-home message for tonight is if I could do one thing, if I ran the world, what would I do? I'd make sure we had good networks for communicating what's happening in different species and the questions and concerns about what might be happening in others that are relevant both to animals and to man. It's clear that man has sort of taken over the world. But that being the case, we not only have the opportunity, we have a responsibility. And we need to look at that responsibility as it relates to animals. And as a veterinary pathologist, that's where my world comes into place. So that's what I have. I think I'm going to turn things over to Dr. Burt.
Thank you, Dr. St. Ledger. And I apologize for switching the roles here. Our next speaker, Dr. Nicole Burt. She is the curator of human and health and evolutionary medicine at the Cleveland Museum of Natural History. She's also adjunct assistant professor in the Department of Anthropology and in the Department of Bioethics at the School of Medicine at Case Western Reserve University. Dr. Burt.
Thank you for your introduction, but also for sticking with us and learning about all this cool stuff. So I am not a co-author, but I was really excited to be invited to take part in this panel because I think it's really important to think about how to translate this type of work to the public, which is what my specialty is. We also heard about a lot of whale people. I'm a human person. So as was mentioned, I'm the curator of human health and evolutionary medicine at the Cleveland Museum of Natural History. And I'm a biological anthropologist. So what that means is I take a biocultural framework to understand human health as both a product of our biology and culture. And that can mean a lot of things, right? So the biology is the easy stuff in terms of your immune system and your anatomy and things like that. But the culture is, I think, what's really cool to this aspect, and well, the cool part of my work, but very applicable here, which is things like medicine broadly, but also things like trying to live close to animals, rehabilitation centers, how we view animals, how we view medicine is all part of that interaction and is something that I focus on, right? So within that research, I generally focus on human diet as a biocultural phenomenon to try to understand and devise intervention around food access and obesity. That might seem like, what does that have to do with any of this? Why are you here? But I really think that approach and the evolutionary approach is a really great lens for thinking about this as how this applies to your life, right? So it's really crucial to understanding health and the future of our planet in the times of climate change, et cetera, by understanding the evolutionary process and how humans fit with biodiversity. And so that's how I kind of frame this kind of work. You might wonder, maybe you didn't, probably you did, what is a curator and what does that mean? So curators are like a professor, but instead of teaching college students, we teach the public and do research, right? So when I say the public, I mean for real the public. So I have talked about some of the themes of this class to people who are 5, and I've talked to the themes of this presentation to people who are 90. And that's the kind of work that's done, and trying to translate it and make it accessible for everyone, because it's that important, right? Like, this is the type of science that is important, everyone needs to understand, even if sometimes it can seem a little like technical or specific for your normal life. So if you're thinking, I've visited a lot of museums and I've never heard of a curator of human health and evolutionary medicine before, what does that mean? You're right, I'm one of 4, at last my count, in the country, health curators. But what we're really trying to do, and we're actually working on designing new galleries here at the Cleveland Museum of Natural History, so anyone who is still local, our Centennial campaign is all about having galleries that really focus on this topic, which is how human evolution, climate change, and biodiversity are all connected, both in the past and in the present and in the future. And so how do we activate people around those themes in a meaningful way? So evolutionary medicine is really just the approach of adding evolutionary biology to our understanding of medicine, which it should already be a part of, but it's not something you necessarily think of in the front of your mind all the time if you are a medical doctor or someone who works in the medical field. But it's really important to differentiate things like, what is happening, what is the virus doing, what is your immune system doing? It's understanding both how, say, something like a coronavirus would evolve, but also how we're evolving in response to that. And the particular framework, and I don't think anyone's used this specific term yet, but I think this is what we're all kind of talking about here, which is the One Health approach, right? So the CDC has a lot on this on their website. So if you're super interested in One Health approach, you can look that up on the CDC website. But the One Health approach is really just an evolutionary approach, right? It's thinking of humans as animals, which we are, as connected to all other biodiversity on Earth and having a lasting impact both on their health and our health, right? We talked a lot about zoonotic diseases. A lot of infectious disease is zoonotic, and there's a lot of transmission back and forth. And so this framework is really important, not just for academics doing research and getting out of our silos, but also in terms of action and policy, right? Because when we're thinking of policy that supports human health, we really need to be including sustainability and biodiversity action in those plans, right? So you can't just conserve human health. We are connected globally, and there's tons of it. You've seen my co-panelists talk about this at length, right? How connected we are to the natural world. So we sometimes feel very separate from it because we're all in our houses right now, trapped. So you can feel very separate from the natural world. But there are tons of human and animal connections every day. All of us live in very close contact to animals. So it's just really important that when we're thinking about being active in our health, we include animals and thinking about biodiversity as part of that model. One of the things I really liked in the paper and that really highlighted to me was the focus on how infectious disease is not a new issue for humans or a new hurdle for human health. And when you really look at human evolution, there's many instances of infection and pandemic being major shapers of human populations and distribution, and not just humans, right? So as you study climate change in the past and over time, things like the ice ages, we have tons of fossil evidence and other kinds of evidence that really show that periods of unstable or unpredictable climate also lead to new diseases, disease spread, which can lead to extinction, as well as something that hasn't really come up in this talk yet, which is that the vectors, whatever is transmitting the disease, move, right? So you might be distant from something now, but as climate shifts, those animals that are normally in warm weather might move up into your range. Things like Zika are a big factor of that, right? So what mosquitoes can carry it, where do they live? And so these are all really important practical things we should be considering. And I don't think we usually think about evolutionary stories as practical, but these are like practical examples of how to think about life. And of course, I know some of my co-panelists have already brought this up. One of the things that we do from a health perspective to stay safe is model. And so modeling is really important, but you can't model things that don't have consistency to them. So as things get more and more out of whack in terms of temperature expectations and what we have data for, we're going to get worse and worse at modeling. I also wanted to bring it into kind of a really local Cleveland example in terms of like, how do people understand and deal with zoonotic disease in their everyday life? Some of you may know, hopefully all of you know, that the Cleveland Museum of Natural History actually has live animals as well as our curated collections. Our live animal collection is all native Ohio species. So not only do we have them at the museum, but you have them in your backyard, right? Coyotes, bobcats, porcupines, also raccoons. I'm sure everyone's seen a raccoon in their backyard. They're all there. And one of the notices we have is that people have to keep wearing their masks when they go out into our wildlife area. And at first we were getting a lot of pushback from visitors, right? They're like, I'm outside, I don't need this. And it's like, yeah, you know to stay away from the people, but you aren't staying away from the animals. And those animals are in direct contact with their caretakers. And because of the nature of how you have to feed a lot of animals, a lot of our animals, they're all, none of them can be released into the wild. They're all have some sort of disability that would not allow them to live on their own. And because of that, they take a lot of personal care. So we really are trying to prevent that loop of transmission. And so that's been a big lift on our shoulders, is teaching people more about zoonotic disease and educating people about how this is actually a practical thing that affects their lives in terms of disease spread and being prepared. And a few of us have mentioned different types of takeaways, but I think my takeaway really is, if we're going to have meaningful and successful public health interventions, we have to think a little bit more broadly in terms of the science and the applicability of what we're doing. and really, as citizens, need to be active participants in public health. Public health isn't something that just comes from on high down. Everyone has to be self-educating about what they can do for public health concerns and doing their best to live by that and expand their horizons a little bit in terms of what they're thinking about. So thank you very much for that brief introduction. I hope you have some exciting questions during the question period.
Thank you. Thank you very much. What an excellent presentations we've heard from everyone. At least for me, it was an eye-opener. We tend to sometimes just be in our own world and surrounding, and we don't think about all those other things that all four of you have just talked about. It was a wonderful presentation. Thank you. We do have a few questions from the participants. The first one is for Dr. St. Ledger. What kind of symptoms would an animal infected with SARS-CoV-2 express? And are the symptoms similar to those found in humans?
So that's a great question, and thank you for asking it. An important thing is we suspect that SARS-CoV-2 likely in marine mammals, where we know we have the binding enzymes, could easily present with the respiratory type diseases we see in people. But we also suspect we could see sort of the systemic conditions that have been seen in people as well. So things like brain problems or encephalitis, muscular problems or myositis. And when we talk about these long COVID infections, remember, one of the concerns is how your immune system interacts with the infectious agent. And we don't really know much about that. What we do know is that these gamma coronaviruses, so there have been gamma coronaviruses not similar to COVID-19, but other coronaviruses that cause all sorts of concerns in marine mammals, dolphins, beluga whales in particular. Those coronaviruses have been associated with acute liver disease and liver necrosis, as well as respiratory disease. So unfortunately, COVID-19 could present in a number of different ways in marine mammals, just like it does in people.
We have a few more questions. Question for Dr. Bird. Are museums around the world adopting department heads with similar titles to yours, Curator of Human Health? And do aquariums and zoos also have these department heads?
So I can speak the best to the US. Like I said, in the US, the last I counted, there were 4 others. At one point in time, 3 out of the 4 of us were named Nicole. I don't think that was a requirement of the job, but it seems to be. And I think almost, so I've had my job for about 6 years. I think the 2 new ones were in the last year or 2. So I think we're seeing, it's not a lot of a curve data points, but there's almost an exponential growth there over the course. So I do think you're seeing more people. There are a little more integration with humans in natural history museums globally. It's kind of piecemeal, but it does have a little bit to do with kind of our history of understanding humans as animals, right? So as someone who studies humans, I would say humans and other non-human primates as an example, or, animals and then, non-human animals, things like that. And so that's newer in the States in terms of how we talk about it, but it is growing. Zoos I can't speak to in terms of position, though a lot of zoos do research, as you can see by some of our colleagues on this call. So I would not be surprised if that was the case.
Thank you. Question for Dr. Ryder. How do you envision the frozen zoo changing the way animal biology is conducted in the next 20 years?
Well, thank you for the question. I think there are enormous opportunities if we're now that we know that these cells that are banked can be used to clone species, Dolly was the first, but now a dozen species at least have been cloned from cells like we bank in the frozen zoo. And now we know that cells like we bank in the frozen zoo can be induced to become stem cells. It can make any cell in the body. And they're just new possibilities that open up. The first is that stem cells are immortal. They can divide and divide and divide and divide. And as we bank this material, we can use it we can contemplate if the science, the requisite science to be able to clone across all species we might want, or to be able to have cells make induced pluripotent stem cells from a wide diversity of animals. And we can use those for genetic rescue, like I showed the Przewalski's horse, Kurt, who's is now approximately 2 months old. If we can intervene at that level, we can substantially reduce extinction risk. We can reduce the loss of genetic variation. And as we bank cells, we are undertaking something that's revolutionary, because it's only been in the last 65 or 70 years that it's been possible to take living cells and freeze them and then thaw them and have them fulfill their function, to function normally, to put them into suspended animation and then to resuscitate them. That's a milestone in the history of life on Earth. That's the first time in 3.8 billion years that it's possible for living material to be present and utilized after an individual dies or a species goes extinct. And if we take a step back and take a larger perspective, that means that we can be accumulating biodiversity, not just watching it disappear. And that's an unheralded and remarkable concept for the role of humankind.
Thank you. We have another question for Dr. St. Ledger. What do you see as the biggest viral threat to the world water systems and their associated wildlife?
Wow. Ask me an easy one, right? I guess my concern, and I suspect my panelists might share this concern, is that the biggest concern that as humans, we think we run the world. So we can employ slash and burn agriculture. We can invade the habitat of woodland creatures in our proximity. We can keep exotic animals in zoos and aquariums. We can essentially do whatever we want. My concern is that in this pursuit of doing whatever we want, we lose sight of the fact that we're only one element of the world. And as a veterinary pathologist, I see it because my focus is in the health and vitality of animal populations. And in the grand scheme of things, when I see humans as one of the biggest challenges to animal populations, it makes me quite sad. I'd be interested to hear what Dr. Nuia thinks specifically towards humans and narwhals. Dr. Nuia?
How we impact them?
How we impact them and how we perceive them or revere them or don't revere them, as the case might be.
Well, the one example that would come immediately to mind is seismic testing. That was an initiative that began to start in looking for oil and natural gas exploitation in the Arctic, which for those in the audience that don't know, if billions of gallons of oil are associated with the Middle East, there are trillions of gallons in the Arctic. So there is a wide use of seismic testing, which have air cannons that produce about 250 decibels each. The average array is 750 decibels. For those of you who've had one of those horns that are at a sporting event, those air horns, and you've had one, behind your ear kind of thing, that's at about 60 to 70 decibels. But 750, and they were doing this basically every 10 to 15 seconds, all day long, all night long. Chris Clark, who some of you may know, was the founder of the Bioacoustics Lab at Cornell, has done maps of kind of the whole ocean and what we are just producing in sound. And keep in mind that most of the aquatic life, its world and its analogous sensory organ to our eyes are their ears. And we are literally drowning out the ocean with our man-made produced noise. Even though the Inuit in Clyde River protested this, it was overturned in the Canadian court, and they had less than a 10% chance in the Canadian Supreme Court, and they actually won. So there is currently a ban on seismic testing, but this will be something that'll be revisited, throughout time. There's a movie in production now about this called The Narwhal's Wake, which will probably be coming out in the next year or so, telling this unusual story. But I think your point is well taken, Dr. St. Ledger, that we need to find ways to live in our world. This isn't a concept of how we reap natural resources and how we just shop the world for kind of what we want, that we have to understand that we're just one element, as you say, in it.
Can I piggyback on that for a second? Just to say that humans are also the biggest threat to humans, not just to animals. And I don't mean that in like a direct violence way. I mean that in a causing our own health and issues and things like that, right? Like we are creating environments that aren't great for ourselves to live in as well as animals. And so putting that out there as this really is a human problem as well as an animal problem, and that you can't escape environment. So from that environment as a key to evolution frame, we might feel like we escaped it with technology and building houses, but we didn't. Evolution's still happening. You're always in the environment. You're always susceptible to natural selection regardless. So it's still happening whether we want it to or not. So that's my 2 cents.
Wow, you're a breath of fresh air, because let me tell you, what you're saying is that as humans, we're the greatest threat to ourselves.
Mm-hmm.
Damn it. As a veterinarian, I thought it was just animals, but no, thank you, Dr. Burt.
All animals, including us.
Wow.
And actually plants too.
Okay, that's a good point.
Well, Dr. Fadul, what do you have left over the fact that we've identified as a species, we've traced the globe?
I have the best question left now, and that's for Dr. Nuia. Dr. Nuia, how do you bridge the world of being a practicing dentist and conducting research like this?
Oh, well, that's an interesting question. And I've certainly been asked that before. Oftentimes I get introduced at a National Science Foundation talk, and when they have to announce that the next speaker is a dentist, everyone is kind of scratching their head. I have always seen the world as an integrated thinker. I see everything as being connected, and I can't help myself. So I even have a hard time when I went through my studies in academics, seeing disciplines as isolated silos or frameworks. I see everything as interconnected. I think we have an immense amount to learn from each other and breaking down those barriers. And interestingly enough, I found indigenous people around the world already have that mindset. You know, we've lost that. I think it was Albert Einstein that once said that it's amazing curiosity survives a formal education. Well, I'm testament to the fact that it can. I've always protected, I think, the inner child within myself and understood at a very early age that in order to keep my personality and me intact, that little kid had to survive. And that's what I bring to everything I do. I make sure that child is alive and well. And for all of you out there who are investigating and, you know, understanding that research has barriers that you can cross, I'm a perfect example that you can do anything with any background. I was an English major in college.
When I agreed to do this, I knew I was going to get an education. I didn't know what a great education I was going to get. That was really outstanding from all of you. So thank you very much for your presentations. I am sure everyone enjoyed what we heard and learned this evening. So again, thank you to all of our panelists. Thank you to the Case Western Reserve University School of Dental Medicine Alumni Association. And most importantly, thank you to our participants for being with us this evening. Before we conclude, I just want to remind the audience that they will be receiving a survey to complete. Please make sure you complete it because it will help provide us with ways to improve our programs and provide for meaningful programs in the future. And with that, I want to thank again all of you. Stay well and be safe. Thank you.